Started. Hello everyone and welcome to today's webinar hosted by Simon's VIP Connect. Thanks to everyone for registering and attending our webinar, and we hope you find it useful for your family. My name is Audrey Bibb and I'm one of the genetic counselors at Simon's VIP Connect. I'm joined today by our speaker, Dr. Elliot Sherr, who is the principal investigator of the Emmigee core of the Simon's VIP research study. Andrea Paul, another genetic counselor at Simon's VIP, will also be helping out with our webinar. Andrea, did you want to say hello?
Hi, everyone.
And today's presentation, as you can see, is entitled Brain Connectivity, What Are We Trying to Learn and How Might It Help Individuals with Autism in 16p11.2. Dr. Sherr will first take us through some of the tools that can be used to tell us how the brain is connected in typically developing individuals as well as people with autism. He will then discuss what findings can be seen on imaging, with individuals with 16p11.2 and what has been found in other populations. Finally, he will talk about how imaging findings might help us to develop new therapies. The presentation will last about 40 minutes and will be followed by a 15 to 20 minute question and answer session. Please feel free to submit questions during the session in the chat window on the left side of the screen. We will try to get to as many questions as possible, but please remember we cannot answer specific questions about medical management for any child. This webinar is being recorded and it will be posted on our website if you or other family members would like to view it in the future. And it's now my pleasure to introduce our speaker, Dr. Elliot Sherr. He received his undergraduate degree from Stanford University and his MD and PhD from Columbia University. He is currently an associate professor in neurology and pediatrics at the University of California, San Francisco, where he directs the Brain Development Research Program and the Comprehensive Center for Brain Development. He sees children in clinic with various differences in brain development as well as metabolic conditions. Welcome, and we'll go ahead and get the presentation started.
Audrey, thanks very much for the introduction, and welcome everyone who's on the call. Thanks very much for taking the time today. And as Audrey suggested, I'm going to talk today about brain connectivity. We obviously haven't studied brain connectivity yet in individuals with 16p11.2, and so what I'm going to do today is show you work that the laboratory has been doing, talk about the kinds of tools that we use, so you can get an idea of what we're going to be trying to learn, and for families that come either to UC San Francisco or to one of the other sites, you'll have an idea of what's going to actually happen. And I definitely look forward to taking your questions. One other thing I want to mention before starting is that the foundation and myself and some of the other investigators were talking yesterday, and this lecture today is going to focus mostly on imaging, but I'm also a child neurologist and will probably host another webinar somewhere in the near future when we can talk more specifically about issues related to child neurology. So I apologize if we're not getting to those today, but we'll definitely be getting to them soon. So I thought following up on Audrey, just to give you a little background on myself, you've obviously heard about Stanford and Columbia. I then went here to UCSF where I did training in pediatrics and in neurology. And I'm a child neurologist. So in that capacity, I see children principally with epilepsy and autism, because those are the areas that I'm well versed in. And if I can say I'm an expert at anything, it would be that I am in diagnosing and caring for children who have anatomically visible brain changes. And I'm going to show you some examples of that in the webinar today. I also run a research laboratory which studies both imaging and genetics. I live here in the city. My wife, Linda Rubenstein, is actually also in the biotechnology field, which is a kind of a funny coincidence. And we live here in San Francisco with our 3 kids. Everybody asked to see photos. I know you can't see me while I'm talking, so I just thought I would show you. We like to travel, so In the bottom right, you can see my family and I at Crater Lake, which is at the border of California and Oregon. We were there this summer. And up above, my wife is in the far right, and I'm on the far left. And we were with a hiking group in Central Europe this summer. So outdoors is something that we like to do when we have free time. So let's move on to the talk itself. This is the basic outline, and I'm going to use a fairly simplistic analogy to sort of explain what I'm talking about, and I apologize if that seems overly simplistic. I hope it's actually helpful. We're going to talk about MRIs and Emmigees and just some of the very basics of them. And then I'm going to refer to brain structure as kind of like assembling a computer, assembling the central processing unit. And then I'm going to break down the different ways that we can visualize the brain and visualize its function. And so we're going to talk about structural connectivity, and so that would be analogous to circuit wiring and on a circuit board for a computer. Functional connectivity we would call the firmware, and I'll show you some examples of that using the tools of Emmigee, which stands for magnetoencephalography, and fMRI, which stands for functional MRI. Then I'm going to talk about things that we can assess that we call task-related activation, meaning what parts of the brain are active when you perform different tasks, like reading words or seeing pictures. And I would say that that's analogous to computer software. And then in the end, obviously, I want to leave time for questions, so don't hesitate to ask. Okay, so if individuals are going to be participating in the study through the Simons Foundation, they will, if they're able to hold still enough to get imaging, will get imaging at the initial site. So there are 3 sites that you can come visit as part of the study. That's Harvard, Baylor, and the University of Washington. You can, when you go to that site, both cognitive and behavioral testing will be performed, and that aspect of the study is going to be directed or is directed by Dr. Ellen Hansen out of Harvard. One of the other things that you also do is obtain a complete neurologic examination and obtain what we would call a structural MRI. So that would be an MRI that you would get if you went to your doctor and had a headache and requested that a brain MRI be done. And so actually you would be provided with the picture of the MRI to take with you on a CD, as well as a report issued by the radiologist at one of the 3 sites. So for individuals who are able to hold still and get an MRI, they will be invited either to come to San Francisco, where we are, or to the Children's Hospital of Philadelphia. And when that happens, the more complicated imaging would take place that's listed here, the MRI with diffusion, functional MRI, and Emmigee or magnetoencephalography. And I'm just going to refer to these in their shorthand. So I might call this diffusion, or I might say DTI, diffusion tensor imaging. Functional MRI I might refer to as fMRI, and magnetoencephalography we'll refer to as Emmigee. So that'll just make it easier so that we can speak in shorthand. Okay, so let's just move along. It's always good to show pictures of what's actually going to happen, and so this is a picture of an MRI machine. If none of you if you have not seen one before, and you can see that on the upper left there's an individual lying down, and that there is a little apparatus which is going over his head, and that's actually the place that's helping you take pictures. If you then look in the if you can then look in the lower right, you can see that there is a apparatus, and then in that apparatus on the upper part of it, there is a set of mirrors, and that allows you, while you're lying down, and this table here will slide into the tube, while you're lying down, to actually see images that are projected on a screen. So for instance, while structural scans are being performed, you can be watching a video, you can bring your favorite DVD along, or all the sites have videos that you can watch. And I actually stuck my own head in the scanner to do that, and I brought my own DVD along, and it made the experience much more enjoyable for me. And then we will also project images for people to look at to answer questions. So that's one kind of apparatus. That's the MRI. The next apparatus is called the Emmigee, and that is a way of measuring brain waves. So it's actually measuring functional activation in the brain. And you can see there are two pictures of people, one where the person's head is actually inserted in the sensor. So you're basically either seated or in a reclining position, and this thing that looks like a hair dryer from a beauty salon in the 1950s is placed over your head. And you can see as that's lifted up that the person has on little electrodes on the head, and those electrodes help in detecting brain activity. And so you sit inside of this machine. There is nothing that hurts or nothing that sort of interacts with you. You see pictures, and you press buttons and speak in response to various tests. And then what's measured is brain activity, and you can see here the pictures of the brain, and there are blue sort of, it looks like one of my children when they were very young took a crayon and just sort of colored in, but actually what that represents is activation in different parts of the brain. And I'll show you an example of that in just a moment. Okay. And here's an example of what's going to happen is that we're going to try to understand where the brain is active and what it's doing when it's active. And you can see here that there are different areas of the brain that are active, and they're pointed out here. So for instance, this is the frontal eye field, which is in the far left picture that's coded in red, and it says right FEF. And we also have other regions, say on the far right side of the figure, it says right DLPFC, right dorsolateral prefrontal cortex. So what we're able to do with both spatial and temporal resolution, so knowing the location and knowing the timing, we can see what parts of the brains are acting up or being, doing the work, and we can actually create videos of this kind of brain activation. And later on in the talk, I'll show you one of those videos and try to explain how that's important for us. Okay. So I mentioned early that brain structure is kind of like saying how is the actual computer assembled. And a structural brain MRI allows you to look at lots of different brain regions. We can assess the size of regions. We can assess the shape of regions, and we can assess their integrity. Are they there or are they not there? Okay. Now we can look at the brain. We're not able to see the brain in sort of a holographic 3D representation. And so the way we view the brain is as if the brain is sliced up in different sections. And obviously the brain's not sliced up, but that's just the way that it's visualized. And you can see in this diagram here that you can visualize it in 3 different planes. So you can visualize it in an axial plane, which you can see written in the lower left side of the slide. You can see it written sliced in a sagittal plane, which is the top slice, and you can see it in a coronal slice, which is in the bottom right. And I'm going to show you examples now of brain images that take advantage of these different ways of looking at the brain. Okay, so here we have two views. On the left, we have a coronal view, and you can see a couple things. One is that you can see that at the rim of the brain that it looks as if it's sort of jagged, and that's what's called the cortical surface, the actual cortex, the outer rim of the brain. And then you see you see a structure in red, which is called the corpus callosum, or I guess it's more sort of in a pink. And you can also see that structure on the right in what's called a sagittal view, as if you're going right down the middle of the brain, sort of in between the eyes, sort of transecting the nose. And you can see an example of that on the right here. Okay, and so these are diagrammatically representing how the brain looks on an MRI. So let's go ahead and look at an actual MRI. And this is a sagittal view again, so the left side of the picture would be where the face and the nose are, and the right side of the picture would be where the back of the head is. And you can see in the middle is a white structure that is called the corpus callosum, and I've highlighted that with the two arrows. And that's something that can actually be either missing or differently shaped in a number of people with autism, and it can actually be missing or abnormally shaped in individuals who have 16p11.2 either deletions or duplications. So that's something that we will be looking for when we do brain imaging on the study participants. There's another area of the brain that's highlighted there that's called the cerebellum, and that's also sometimes abnormal. There are lots of other brain regions. I'm just pointing these two out because they're very easy to see visually, and they make the demonstration of the kinds of findings that we might observe when we do a structural MRI. Okay, so now remember the normal corpus callosum that you see well displayed here, and we're going to look at examples in the next slide of abnormal corpus callosa. And so if you look at this picture here, there are 4 images. All of them are, again, sagittal images of the brain, 4 different individuals. And the one in the lower corner is a normal one. It's the same image as in the prior slide. And if you look to the upper left image, there is no corpus callosum. So that white structure, which is actually hundreds of millions of axons that cross between the 2 halves of the brain, is missing in this individual. And some individuals don't have it completely missing. They just have part of it present. And that's evident here in the upper right figure, where you can see partial, and there's an arrow pointing to that partial structure. And then you can also see in the lower right that sometimes the corpus callosum is there, but it's very, very thin. And that's represented in that slide as well. Okay, so these are just examples of the kinds of structural changes that we may see on an MRI. And so myself and other members of the imaging team will look over all of these images very, very carefully. So each person who gets a structural brain image, as I said, will be getting an official report from the radiologist, but we'll also be looking at all the participants to see if there's any commonalities and to see whether some of these brain changes help us understand the degree or the spectrum of problems that 16p patients may face. Okay, so let's move on to a different kind of imaging that we call diffusion tensor imaging, or DTI. And what we're seeing here is what I would refer to as the circuit wiring. So the brain is composed of lots of different nerve cells, many, many millions of them, and they have a cell body, and then they have long structures that are called axons that connect from one cell to another. And that's how the brain gets connected up. And some of those axons go very short distances, but some of them go very long distances, very much like circuit wiring. And very, very brilliant physicists have come up with a way to use the MRI, to use the magnetic field of an MRI to figure out where these connections are made in the brain. And I'm going to show you some pictures that investigate that very issue. So you might ask, how do you do that from a technical point of view? And so this is what I'm going to try to explain here. It's kind of complicated, and I apologize if it's difficult, but I'm going to try to break it down as much as possible. On the lower left, you see a picture of little circles that are attached together by sort of a jaggedy line. And what that's trying to represent is diffusion of water. And if water is in a glass, or sort of more something that everyone has probably seen, if you have a glass of water and you drop into the glass of water food coloring, you know that the food coloring will diffuse. It'll start out as a big blob in the middle, and then it'll diffuse in kind of a random way until eventually the glass will be a dull color of whatever the food coloring is. If it were red, it'll be sort of a dull pink once it's all diffused. And that's what the radiologists call isotropic diffusion, meaning it's randomly diffusing. The restriction, there's no restriction of diffusion except for the boundaries of the glass, which are very, very large. Now, axons, which are tiny, thin tubes that connect nerve cells to each other, are bundled together almost like you would have either wires in an old telephone system or optic cables in more advanced computer and phone systems. They're all bundled together very tightly, and each one of them has water inside of it. And so what you see on the right side of the picture are, again, the water molecules, and their pattern of movement is restricted by the fact that the tube or the axon is very, very narrow and thin. And so water will diffuse inside of the axon and not sort of pop out. And the magnetic field can measure this water diffusion, which is a pretty significant technical feat. So if you can measure that diffusion, you can then model using mathematics where you think the tracks actually are. So I want to focus your attention on the picture on the lower part for a moment. And so what we do is each little box that you see here represents a little three-dimensional space inside of the brain. And inside of each one of those little three-dimensional spaces, which is actually equal to a little fleck of color in the picture that you see above it, each one of these little boxes, we can calculate the direction of water diffusion. It's not exactly that direction, but that's the overall direction inside that little box. And then we can use mathematical models to draw out a picture of where we think the tracks are going. And you can see examples of that right here where we've color-coded it. So I want to focus your attention on the red structure that's in the middle, and that's actually the corpus callosum. And we can know now that it is the corpus callosum because it is going from the left side of the brain to the right side of the brain. And we knew that before because we knew that from prior anatomic studies. But we can use this same tool to discover new things about how the brain might be connected. So this is an example here of how in a person who has a normal corpus callosum, you can see it all drawn out. And so again, the corpus callosum are fibers that connect the two halves of the brain, the left side and the right side. And another feature about these tracks is that if there is a fiber that starts out in the very front of the brain, it ends up on the front of the brain on the other side. So if it starts out in the left on the front, it ends up in the right on the front. And you can see that if you look in the figure that's in the middle on the bottom, you can see that there's color coding of the different regions. So in the very front, there's an area that's called BA10, which is an area of the brain. And you can see this red set of bundles that goes from the very front on the left to the very front on the right. Similarly, if you look in the middle, there's an area called BA6, and that's sort of light blue, and you can clearly see that the fibers start out on the left side, in the middle, and end up in the middle on the right side. And so we call that homotopic, meaning that it goes to the same location on the other side. However, we asked whether in individuals who are born with a small corpus callosum, so again, if you look at the slide in the upper left, you see a yellow arrow, and that's pointing to a small corpus callosum. And then we can plot out, using this mathematical modeling that we call tractography, where these fibers are going. And we've color-coded that in this picture right here. It turns out, however, that some people don't have just what we would call homotopic mapping, where it goes from the front to the front or the back to the back. And you can see an example of that homotopic connectivity in the far left set of images. So the top image is, again, a conventional MRI image, and the arrow is pointing to a small corpus callosum remnant. And then the next pictures down are looking at the tracks at different angles. So this is looking at as if you're, the first one down in the middle is as if you're looking at it from the top of the brain, and then the one in the bottom, again, is what we call the sagittal view, and is as if you're looking at it from the side of the brain. And I want to bring your attention to the structure, which is the second one over to the left. And that structure, you can see that there's blue fibers that go from the front to the front in what we would call a homotopic way. But interestingly, there are new fibers that go from the front, and you can see that sort of on the left side of the picture, and they go to the back of the brain on the right. We call those heterotopic connections because they go to different locations. And this is something that would not be seen on a conventional MRI. So if you look at the structure of that, it just looks like a partial corpus callosum on the structural MRI, which is the picture above with the yellow arrow. But we rather see that there's new fibers that are unanticipated in the brain. And this technique allows us to see this kind of connectivity, this changed or different connectivity. And we may see this kind of connectivity in individuals with 16p. Now, what I'm going to do now is show you a video. Oops, I apologize there. Go back. I'm going to show you a video, and for that, I'm going to need to switch out of the presentation and just show you my computer. So hold on just a moment. I'm going to make those changes. Okay, so now you should see in front of you a picture of different fibers, and they're color-coded, and I'm going to show you a movie that shows different kinds of connections in the brain in this residual corpus callosum. So these are all the fibers, and you have the left side and then the right side. And then we're going to look at ones where they are homotopic, where they go from the same location to the front and front, sort of middle front to middle front, and back to back. And now we're going to show ones that are not normal. So this one is in the left bottom on the right, I'm sorry, and then the upper part, and then this is frontal on the left, and posterior on the right, and posterior on the left, and frontal on the right. So these are different than the original ones. We call those heterotopic connections. And then here, all the fibers in color coding, showing it from left to right. So this kind of connectivity is only able to be seen using this tool called diffusion tensor imaging. Okay, I'm going to return to the regular presentation. Okay, and move on to the next slide. One of the other things that we can do using this same tool is measure the size of some of the connections. So what you see there are 6 panels here. In panel A and B, we're drawing a white circle around a particular connection. A particular connection in the brain that's called the cingulum. And we draw these 2 arrows, and then the computer helps us connect these 2 circles. And so if you look in C, you see there are 2 white lines, and those white lines represent where those circles are drawn. And you can see not the upper structure, which is the corpus callosum, but rather the lower red structure is a track, the cingulum, that's drawn using this mathematical model. And then down below in D and E, we're trying to measure the same track, but we're measuring it in somebody who's born without a corpus callosum. And you can see that that track, that lower one, is very, very diminutive by comparison. And so again, we use this tractography, this advanced imaging, to see the different connections in the brain. So we can not only see abnormal connections in terms of location, but we can also see smaller connections. And this is also important, as that'll help us understand in a better way how the brain is connected in individuals with 16p. Okay. So I've talked a little bit, again, to use sort of the computer analogy. I've talked about computer structure, and I've talked about the circuit diagram, how things are connected. But how are they connected in a functional way? And I'm going to use the sort of the crude analogy of calling that firmware, not quite hardware, not quite software, sort of in between. And what you see here is just a diagram of different regions of the brain that are all connected by these lines, and each little dot is a place of activation. What we're measuring is what parts of the brain are active at different times. So imagine for just a moment that you have a thought, and if we could imagine that the thought is consolidated in one particular part of the brain, let's say the back of the brain, where that white structure is that has all the arrows going to it, and while that's happening, other parts of the brain, maybe not immediately, but almost immediately afterwards, light up, meaning that those parts of the brain are active. And then we can draw arrows or lines connecting these different regions of the brain and essentially say, aha, that part of the brain is working, and at the same time, other parts of the brain are working. And when those other parts of the brain are working all together, they create a circuit. And that circuit, or that firmware, is what's actually performing the task, whether that task is reading a word or looking at a picture or performing a mathematical calculation. Okay. And so here is an example using the tool Emmigee. And if you remember, the way Emmigee works is that it measures the electrical activity, or actually the magnetic deflection of the electrical activity inside of the brain. And so this is what we call resting state. So that means somebody is sitting in the Emmigee with that 1950s hair salon dryer over your head, and you're just sitting at rest with your eyes open or your eyes closed, just at rest, not doing anything, and the machine is measuring the different connections that your brain is making all the time. And you can measure that over a few minutes, and then you can compare those connections that are being made across a whole group of individuals. And so what this shows is on the left is a group of individuals who are controls, and so that's in figure A. And the color coding shows you the degree of connectivity. So the more yellow it is, or almost into white, the more connected it is, and the more red or dark red down to black, the less connected it is. And you can see just by glancing at it that the group in A is much more active than the group in B. And so A is the control group, and B are the individuals born without a corpus callosum. And so you can see that there's reduced connectivity in the group of B. And then down in C is an attempt to map out the specific areas that are underconnected. So in this case, the degree of underconnectivity is coded by the colors. And so if it's sort of a aquamarine or greenish color, then it's most underconnected. And if it's dark blue, then it's less underconnected, but still underconnected. And you can see lots of different areas that we've circled and labeled showing that different parts of the brain are underconnected. I'm not going to go into the specifics of which parts of the brain are underconnected, but just to tell you that it's important for us to know which parts of the brain are connected, because lots of other literature, lots of other scientists have been working on these different areas of connectivity to help us understand what parts of the brain participate in what kinds of tasks. Okay. There is another tool that's called fMRI, or functional MRI, and we can do a similar kind of analysis. It's slightly different, but it allows us to look at different regions of the brain that are connected. And so what you're seeing here is exactly that, different connection networks inside of the brain. And it's, some of you may have heard of the term BOLD, and BOLD stands for blood oxygen level degree. So it's the degree of blood oxygen levels. And that allows us to measure activation in the brain. So both of these, the Emmigee and the MRI BOLD, are non-invasive measures of how the brain is connected. And here's an example of underconnected brains in, again, a patient group. And you can see we get very, very specific regions that light up. It's not as if the whole brain is lighting up. And that allows us to focus on understanding what these different degrees of disconnection may mean for someone. Okay, so let's actually move on to that. I've shown you different connection problems, but what does that mean? What are the sort of the real-world implications of saying that different parts of the brain are less connected? And then something to think about is there something that we can do to enhance those connections, and does that mean something? And I'll come to that in just a moment. Okay, so this is a task that you can do when you come visit, and it's called a Tower of London. And the idea is you can see that there are 1, 2, 3, 4, 5 discs that are stacked up from the biggest at the bottom to the smallest at the top. And you're shown a picture of, let's say, the simplest version would be the smallest at the bottom and the biggest at the top on the peg on the right. And you would have to figure out a strategy of moving the discs from the left to the right. You're not allowed to take off more than one at a time, and you have to put the one that you take off on one of the pegs. So you can put it on a middle peg if you want to create an abnormal design, or if you're just going to invert it, you would obviously start from the top and work your way down. So what this does is this tests your ability to visually understand things and then to create a strategy for solving the problem, because you have to come up with a strategy of stacking these disks to create the picture that you're shown. And we call this kind of skill executive function. And another way to think of executive function is organizing yourself during the day. Can you, fill out your calendar and go from place to place and do your shopping and complete your tasks at work? And that would be sort of an analogous real-world demonstration of executive function. But we can measure it in a timed way here at UCSF or at other institutions, and we can compare this kind of test to how a whole group of neurotypical individuals do on that. And so we've done this kind of test on a whole group of individuals, and then we ask, does how you perform on this test correlate with how different parts of the brain are connected? And so that's what we're demonstrating here in this slide. Let's focus on the bottom part of this picture for just a moment. And you see in the bottom part of the picture, there's a circle around an area, and the legend says left BA8. So left Brodmann area 8, that's just a way of telling us what address that is in the brain. And that's a part of the brain that is important, previously known to be important for executive function. And what we can see here is that the black line on the right is showing us that there's a correlation in individuals in our study cohort, individuals with autism, showing us that the degree of connection correlates directly with how well they perform on that task. So connectivity has a direct correlation with performance. Okay. So we can do the same thing with other tasks. So this is a task that sometimes is called Stroop interference, but we can also use it for very, very simple tasks, which is if I ask you, start at the top left and read the words. So you would read red, blue, orange, purple, orange, blue, green, red, blue, purple, green, red. So just doing the first 3 lines. Then I might also ask you, read the color. So then you would say red, blue, orange, purple, orange, blue, green, red. Those are simple. Then the last 4 are more complicated. Green, red, purple, blue. And that's actually in contrast, and that's more difficult. But if we just ask you to read the first 2 lines, those are congruent, and they're easy to do. And so that's a way of measuring how your brain can process language, as well as, again, processing the colors. And just like in the tower test previously, we found that the speed with which somebody could read those words or read the colors activated different regions in the brain. And so we see here both the left MTG on the top and the left STG. And both of those areas are the part of the temporal lobe, or the temporal gyrus, medial temporal gyrus for MTG and STG for superior temporal gyrus. So those are 2 different parts of the brain that are involved in language. And so we were not surprised to see, but we were struck by the fact that the degree of performance correlates very, very tightly with how connected those language areas are. So connection in the brain correlates very tightly with performance. More connection correlates well with better performance. And we can do this not only with what we would call executive function or language tasks, which are things that are important for individuals with autism, but also we can ask that similar kind of question in testing individuals' ability to understand emotion. And so you can see here we've labeled 6 different faces, and we've labeled the faces here with the kind of expression that goes with it. So the first one is happy. You can see the individual is smiling. The next person is clearly sad. The third person looks like he came out of a bad Hitchcock movie, struck with fear, etc. And what we do is we show individuals these faces, but without the descriptions, and we can ask them, what is the facial emotion, and try to gauge someone's ability to understand facial emotion or emotion generally. And here's another example where emotional cognition or understanding emotions, again, correlates with performance, or rather the performance of understanding emotion correlates with connectivity in a different region of the brain. And that's highlighted with the blue pictures in the brain on the right side. Okay. Now let's talk about not just connection, but actually performance of tasks. Now I mentioned again, the connections are kind of like the firmware, and the tasks are kind of like the software themselves. So let's look and see what we can learn by doing that kind of approach. And so this here is something that's called an auditory verb generation task. And you can see in the picture that you're presented with a word. You're presented with the word car. And so you hear that, and that's at the beginning. And then once you hear that, you have to process that word in your head, and then you have to come up with an answer. And the answer that the person gives is drive. And so you say car, you hear car, and you say drive. And while that's happening, we're measuring the activation in the brain. We're measuring it from the time that you hear the word car all the way through to when you say the word drive and then slightly after. And we can then look at activation in the brain. We're most interested actually in the in-between period between hearing car and saying drive, because we want to know what parts of the brain are working while you're trying to come up with the word drive. So in other words, you have to hear the word car, so the hearing part of the brain is working. Then the next thing you have to do is interpret the word, so the brain has to interpret that language, understand it, and then you have to come up with a response. So you have to digest it even further and interpret it further, and then pick a new word, in this case drive, and then actually produce the word. Form your lips into the answer. And all of that is happening over, you see, a very, very short time window. So at the top of the picture, you can see it starts out at 0 milliseconds and ends at 850 milliseconds. So that's all less than 1 second. So we all are able to do that very, very rapidly. And we can measure how the brain is working when that's happening. So I'm going to show you a video, and I apologize. The slides, again, are not working perfectly for this, so I'm going to have to go offline and show you what's on my computer. So hold on just a moment. The screen, and you see blue, and blue means activation. And I want to focus your attention on the 3 pictures on the left of the brain and the 3 pictures on the right. And basically what I'm trying to show you is that the 3 pictures on the left are showing you the top, the front of the brain, the side of the brain, and the top. And then the other ones are showing you the back of the brain, the right side of the brain, and the bottom. So you can see the whole brain from different angles. And you can see right now, this is the end of the slide, that most of the coloring occurs on the left side of the brain. So you can see the left side of the brain in the front, So that person is facing you, so that person's left would be on your right. Then the brain is tilted to the side, and that's the left side again in the middle section. And then the top of the brain is, again, you can see the left is on the left side and the right is on the right side. You can see that the left is active. And similarly, when looking from the back of the brain, you can see that the left is active. So this is a healthy set of controls. This is actually summarized data from many, many people. And I'm going to show you the time course of how this is activated. So you can see there's a time clock in the middle, and you can see slowly over time, more and more of the left side of the brain gets active. I won't detail the areas of the brain, but just to tell you that it's the left side of the brain, and almost no activation at all is occurring on the right side of the brain, which would be seen on the brain turned to the right side facing us, which is the middle of the three on the right-hand side. And you can see, in this case, you can see my arrow pointing at it for you. So I'm going to show the video again. Again, you can see the left side is becoming quite active and the right side is not active at all. Now, what if we do this same sort of test in individuals who are born without a corpus callosum, which is one of the groups that our lab has been studying? And you can see, again, looking at the end of the picture, that you have activation on the left side of the brain and on the right side of the brain. And you can see that on the arrows here that I'm pointing, the left and the right side, and you can also see it in the front and the back and the top. And let's look at how that comes on. Okay, so it looks like there is activation in both the left side and the right side, and that the right side seems even more active than the left side, and that the amount of activation on the left side is smaller than the amount of activation in healthy controls on the left side. So indeed, what we're seeing is that there is reorganization in the brain of how the brain processes language. So in healthy controls, the left side of the brain is being utilized. In these individuals, both the left and the right side is being utilized, presumably because the left side is not normally functioning. Okay, I'm going to go back to the slide presentation. So what I've shown you now is a number of different things that we can see by looking at the structure of the brain, by looking at how the brain is connected using diffusion tensor imaging, to understanding the firmware, understanding how the brain is connected, and then understanding how the brain functions when given tasks are put to the person. So why do we want to understand? Why do we want to take all these pictures? Sort of what's the point? What do we gain? And you can see that I'm sort of getting at that at the bottom of the slide. We want to understand brain structure. I mean, I think that that's very important. We want to be able to compare structure and function. I apologize, there's a little typo there. And we want to know how all of that is connected up. Right? We're not designing the computer, to use the analogy, but we're observing the computer and trying to understand how it functions and how we can make it function better. And that's sort of the main point. Can we use this understanding of brain structure and function to help us understand how to help individuals with various cognitive and behavioral challenges? And so this is an example of a paper that was published by a group from Pittsburgh, where they were teaching people who had dyslexia how to read in a better way. And so this is behavioral training. So a group of individuals who were identified with dyslexia were asked to come in, and different training paradigms were introduced. And what they did is that they measured connections, just like the connections that I showed you, in the brain, both before and after the training went on. And so in A, you can see that the areas that are in red and orange, just like the sort of the changes that I told you previously, you can see that it's bar-coded, color-coded rather. So dark red is less change, and sort of yellowish to white is more change. You can see that there's an area of the brain that gets more connected after somebody learns how to read using this new teaching paradigm. So actually the brain connections correlate not only with how people do, but with actual improvement. So more connection correlates with improvement on the task. And B is just showing you that this is the same region that if you compare individuals who don't have dyslexia, so people who read normally, to the dyslexics, it's the same area that's better connected in healthy controls. And that intervening by teaching with new strategies actually results in improvement in that exact same area. So it suggests that understanding connections can actually lead to assessing those changes in a more precise way. Okay, here's an example where people are asked to read the expression, and you can see here that there are 4 choices: interested, jocular, loving, and content. This is clearly somebody who is interested, and we ask individuals with autism to read what's called reading the mind and the eyes, reading the emotional intent behind a facial expression, and specifically just the eyes to sort of make it more difficult. And we know that from work of others that a drug that's called oxytocin, which is actually made in the body naturally, by taking that and inhaling it through like a nasal sniffer, that 90 minutes later, if you're asked to do this test, you can actually detect these emotions better. And so one of the things that we would wonder is, could we observe brain changes after this kind of intervention? And in a similar way, autistic individuals tend to look at not the eyes when they look at individuals, but rather the face, the mouth, or the chin, or the ear. And you can see here, here's an individual on the right, and this is looking at their eye tracking. So it's recording where in the picture your eyes are looking when you're gazing at something. And so somebody with autism is looking at the corner of the mouth and looking at the shoulder. And after that person is treated with oxytocin, they spend more time gazing at the eyes. And so these are examples where understanding connections and understanding the problems with brain functioning would allow us to merge the two together. So merging brain functioning with these cognitive assessments in a way that would allow us to improve and monitor any interventions that we would introduce down the road. Okay, so I've been talking quite a bit here. And I apologize if this has been too much information for you. I'm happy to answer questions. But before I do that, I just want to highlight that there's a lot of individuals involved, both at UCSF and at all the other sites that are participating in this. And I've highlighted their names, and you'll probably be meeting with a number of them when you come visit us in San Francisco. And thanks very much for listening, and I'd be happy to take questions at this time.
Okay. Well, I do have a few questions that I've gotten from the families. Lori was wondering, are benign findings on MRIs typically something that needs to be followed up with another MRI in the future?
Benign findings. If the findings are indeed benign, then it's highly unlikely that they would need to be followed up.
Okay, great. And Hillary had a question. She was wondering, how exactly would the deletion or duplication cause a structural change in the brain?
Right, that's a really good question. We know that in 16p that there's about 600,000 letters of DNA that are in this region that is either deleted or duplicated, and there's a number of genes in that region. There's about 20 to 25 genes in that region. And the fact that I can't tell you exactly how many gives you some sense that we're still learning about this region. A number of these genes seem to play a role in brain development. And obviously there's a lot of investigators around the country who are trying to better understand exactly what those genes are. But we know that some of them play a role early on in brain development. And if there's only one copy instead of two copies, or three instead of two, that the sort of the instructions that the brain is reading as it's undergoing development can be altered, and it can result in these changes.
Okay. And the next question is from Caroline, and she wants to know, can low oxygen saturations cause a concern for future brain problems?
So I guess I would ask for further clarification on what that means
Okay.
by low oxygen saturations before I can really answer that question.
Okay. And I'm not sure if she's actually on the webinar today, so I'm not sure we'll be able to get that, but I can just move on to the next one. Another question from Hillary, actually. She was wondering, she's heard that 16p11.2 duplications can be associated with both autism and schizophrenia, and she was wondering if those two issues are likely to coexist in one person.
So they typically don't coexist in one person. But it is theoretically possible that one person could have both.
Okay. Okay. Well, I think that's kind of all the time we have. We're running a little bit over, so I don't want to take too much of everyone's time. But thanks so much for everyone for listening and for being a part of our webinar. And the next one will be on March 30th. And thanks again for joining us.
Thanks again.